Hello, welcome back. We're ready to deep dive into PureTarget, the PCR-free targeted sequencing with methylation. This powerful method uses CRISPR-Cas9 for enrichment, all without PCR amplification. Let's walk through how PureTarget works. You start with high molecular weight genomic DNA, ideally with at least 50% of fragments longer than 30 kilobases. You'll typically use 1 to 4 micrograms per sample. The first step chemically modifies the natural ends of the DNA to prevent them from being ligated later in the process. Then, the Cas9 enzymes, guided by specific single guide RNAs, designed to flank your target regions, cut the DNA precisely on either side of each target. These targeted fragments, which might be 4 to 5 kilobases for normal alleles, or over 20 kb for expanded repeats, now have defined ends created by Cas9. These ends are then DA-tailed, and PacBio's indexed SMART Bell adapters are ligated on. Finally, a nuclease digestion step removes all the non-circularized, non-targeted genomic DNA. This leaves you with an enriched library of your target molecules ready for hi-fi sequencing. What are the key advantages of this PCR-free approach? First, the workflow is streamlined. You can potentially go from sample to answer in about 3 days, and that includes DNA extraction, library prep, a 24-hour sequencing run, and rapid analysis. But the major benefits truly lie in the data quality, especially when you compare it to methods involving PCR, like amplicon sequencing, or even hybrid capture, which includes PCR steps. Because PureTarget is PCR-free, methylation is preserved. You're sequencing native DNA, so you retain all of the 5-MC methylation information. There's no GC dropout. PCR can struggle with very high GC content regions, often leading to uneven coverage or complete dropout. PureTarget avoids this bias. There's less size bias. PCR preferentially amplifies shorter fragments. PureTarget has significantly less bias against long fragments, which is crucial for studying large repeat expansions. And there are no PCR replication errors. PCR can introduce errors, especially in repetitive regions. PureTarget sequences the original molecule, effectively avoiding these artifacts. To get the best results with PureTarget, the quality of your input DNA is important. Because you're physically cutting out the target region from the native DNA, longer starting fragments mean a higher probability that your target region is intact between the Cas9-induced DNA breaks. We recommend using DNA where at least 50% of the fragments are longer than 30 kb. This is often measured as a GQN at 30 kb being greater than 5. Using high-quality DNA, like that extracted with PacBio's nanobind kits, generally results in higher and more even coverage across your targets. For the standard 20-gene panel, you can expect coverage around 300 to 400x with high-quality input. For analyzing the complex data from targets like repeat expansions, PacBio provides specialized tools. The Tandem Repeat Genotyping Tool, or TRGT, pronounced target, takes the aligned hi-fi reads as input. It identifies the repeat region based on a definition file, determines the sequence and structure of the repeat motif within each allele, and counts the motifs. It also calculates the allele lengths, assesses the methylation status of CpG sites within and flanking the repeat, and provides read depth information. The output includes informative visualizations called target plots and a standard VCF file for downstream analysis. PacBio offers PureTarget as an end-to-end solution, which is particularly demonstrated with the PureTarget Repeat Expansion panel. The workflow starts with DNA extraction, nanobind recommended. It then uses the PureTarget kit for targeting and library prep for 20 clinically relevant repeat expansion genes. It allows for multiplexing of up to 48 samples per run on Revio or Vega systems and includes analysis via SmartLink software, which incorporates the Target tool for genotyping and QC. The off-the-shelf repeat expansion panel uses 1 to 4 micrograms of high-quality DNA. GQN at 30 kb greater than 5 is also recommended. It typically yields over 200-fold mean coverage per target, with a minimum of 50-fold for unexpanded alleles when multiplexing 48 samples on Revio or Vega systems. The resulting library molecules are typically 4 to 5 kb, but can exceed 20 kb for expanded alleles. And crucially, the methylation status is detected. PureTarget provides a robust, PCR-free method for accurately sequencing challenging genomic regions. It preserves vital epigenetic information and enables comprehensive analysis of complex variations like repeat expansions.